Usually, when we talk about a medical diagnosis, there's this expectation of precision. Like you break your arm, the x-ray shows a jagged white line, and the doctor just points at the film and says, well, there it is. It's incredibly straightforward.
Right, and we crave that. We really want biology to be visible, mechanical, and easy to categorize.
But the moment you step into the human brain, that x-ray machine effectively breaks. You are suddenly navigating a landscape that is it's completely mysterious. And a groundbreaking 2026 paper hot off the presses from the journal Immunity by Chen and colleagues, well, it just revealed exactly why the brain plays by its own rules.
It really did. It's a fascinating piece of work.
Yeah. It turns out the security guards inside your brain, your dedicated immune cells, are operating on a completely separate, secret genetic blueprint from the rest of your body. And our mission on this deep dive today is to uncover exactly how that works.
Which is a monumental discovery. I mean, we aren't just talking about cataloging microscopic cells here. We are digging into the fundamental blueprints of human development. Because in biology, if you can understand exactly how a cell is built from the ground up, you can finally understand what it's actually capable of doing.
Exactly. And today, for you listening, we're talking specifically about microglia. If you aren't familiar, microglia are the brain's personal, highly specialized immune system.
They are. And they don't just fight off infections, which is what people usually think.
Right. They act like microscopic gardeners, they prune neural connections, they clean up dead cells, and they literally shape how the brain wires itself. And scientists have essentially just discovered the secret genetic password that creates them.
But to understand why this password is so revolutionary, we have to rewind the clock all the way back to the embryonic beginning.
We do, because we really need to explain every single mechanism here step by step. So let's talk about how blood and immune cells are born in the first place. That's a process called hematopoiesis, right?
Yes, hematopoiesis. And the biggest misconception here is that the body just builds a single factory that pumps out all your immune cells for your entire life. Well, that's not how it works at all.
Oh, really?
Yeah. Your immune cells are actually born in 3 distinct chronological waves as an embryo develops.
3 waves. Okay, let's unpack this. Let's trace that timeline because I know the timing is the key to this whole mystery for you listening. Walk us through wave 1.
Sure. So the first wave is what we call the primitive wave. This happens incredibly early at embryonic day 7.0, which researchers refer to as E7.0.
Wait, day 7? That is incredibly early.
Exactly. Think about how early that is. This takes place in the yolk sac well before the embryo even has a functioning liver or a skeletal system.
Wow.
And this first wave produces the very first pioneer cells, known as primitive erythromyeloid progenitors, or PEMPs.
Primitive erythromyeloid progenitors. Okay, that's a mouthful.
It is, but here is the absolutely vital detail. These specific, extremely early cells from wave 1 are the only ones that ever become your brain's microglia.
Okay, so the brain essentially calls dibs on the very first batch of immune cells ever manufactured, pulls them inside the developing skull, and locks the vault.
That's exactly what happens, because just a day or so later, around E8.0 to E8.5, the body initiates wave 2, which is the transient definitive wave.
The transient definitive wave. What happens there?
Well, the cellular factory shifts from the yolk sac to the newly forming fetal liver. The second wave produces monocytes that travel out and seed the rest of the body's tissues.
Right.
And they actually replace almost all of those early primitive macrophages created in wave 1.
Yeah.
But, and this is crucial, they don't replace the microglia. The brain's vault is sealed.
So the microglia just stay put, completely isolated from whatever's happening in the rest of the developing body.
Right. And then finally, we get wave 3, the definitive wave. This kicks off around E10.5 in a specific embryonic region called the AGM.
The AGM, what does that stand for?
It stands for aortogonad mesonephros. It's just a specific developmental region. But the important part is that this wave finally establishes the adult stem cells that move into your bone marrow.
Ah, okay.
This bone marrow factory is what you and I rely on to produce all of our circulating blood and immune cells for the rest of our adult lives.
Okay, so to map this out clearly for our listeners, the custom immune cells in your brain come exclusively from wave 1. The standard immune cells patrolling your bloodstream and organs mostly come from wave 3. That makes sense.
Yes, exactly.
But regardless of when they are born, they're all still macrophages, right?
Mm-hmm.
They're all immune cells. So there has to be a master genetic blueprint they all share to do that job.
There is, and it's a gene called Zeb2. Zeb2 is what we call a master transcription factor. Specifically, it acts as a repressor. It is absolutely 100% required for any macrophage to exist anywhere in the body.
Let's expand on that concept of a repressor for a second. Because usually when we think of a gene turning on, we think of it building something. But Zeb2's job is to stop things from being built.
Yes, and that is a crucial mechanism in biology. Cellular identity is just as much about what a cell doesn't do as what it does.
Oh, that's interesting.
Yeah, Zeb2 basically sits inside the cell and actively turns off the instructions that would accidentally turn that cell into a completely different type of immune cell, like a dendritic cell or a B cell. It silences the competing blueprints.
So if you don't have Zeb2 functioning, you don't have macrophages. Period.
Exactly. The cell loses its identity entirely. But here is where we hit a massive biological anomaly. In normal, adult definitive blood development, that's wave 3 in the bone marrow Zeb2, requires a highly specific genetic enhancer to turn on.
An enhancer.
Right. It's a piece of DNA known as the -165 kb Zeb2 enhancer.
Okay, let's use an analogy to visualize this. Think of the Zeb2 gene as a vital machine, like the massive central engine that makes an immune cell function. And this -165 kb enhancer is the primary power switch on the wall that actually turns the engine on.
That's a perfect way to visualize the relationship between a gene and its enhancer. The engine is useless without the switch to run the current.
But wait, if I'm tracking your timeline correctly, this presents a massive logical paradox.
It does.
If every single macrophage needs the Zeb2 engine to run, but that specific wall switch is only designed to work during the later adult waves of development, well, how on earth did the early bird microglia from wave 1 turn the engine on? They are locked in the brain vault before the standard wall switch is even built. That shouldn't be physically possible.
And that profound paradox is exactly what the researchers set out to solve.
Okay, before we get to the solution, let's do a quick recap for you listening. We promised to keep things organized. So, bullet point 1, we have 3 distinct waves of immune cell birth, and the brain's microglia exclusively come from wave 1. Right, bullet point 2. All macrophages need the Zeb2 gene, which acts as the main engine. Bullet point 3. Adult cells use the -165 kb enhancer as a wall switch to turn it on. Which leads to our big question for you to ponder. If you were designing the human body, why would you give the brain's security system a completely different set of genetic blueprints than the rest of the body?
It's a great question. And to figure out how the brain actually does it, the researchers realized that the brain's immune system must have a completely different hidden mechanism for turning on its power.
So how do they test that?
Well, they designed an incredibly elegant experiment to see what happens when you intentionally cut the power to the main grid.
Wait, how do you cut the power to a specific gene in a living animal?
You use genetic engineering. They utilized a highly specialized mouse model called the Zeb2 delta -165 kb knockout mouse.
That sounds like a mouthful. Knockout mouse.
Yeah, all it means is that the researchers went into the DNA of these mice and surgically deleted, or knocked out, that specific -165 kb power switch. They left the Zeb2 engine perfectly intact, but they removed the wall switch that turns it on.
And then they just watched to see how the immune system would react.
Exactly.
And the fallout in the adult immune system was catastrophic, right? Without that switch, the adult bone marrow factory essentially shuts down. The wave 3 monocytes and macrophages completely failed to develop.
They do. But the researchers didn't just look at the bone marrow. They looked at the tissue-resident sentinels, the immune cells that live permanently in organs like the liver and the lungs.
And the state of those cells was pure chaos.
Absolute chaos. Because they lost the normal Zeb2 regulation, the repressor we talked about earlier just wasn't working. The engine was off. So the genetic instructions inside these cells completely short-circuited.
I remember the paper goes into deep detail on this using RNA sequencing. Let's explain that, because RNA sequencing essentially lets scientists read the biological receipts of what a cell is doing at any given moment, right?
Yes, RNA-seq looks at which genes are actively being translated into proteins.
And when they read the receipts of the surviving macrophages in the liver and the lungs of these mutant mice, they saw a massive identity crisis. Genes that normally build entirely different immune cells suddenly flickered on.
Right, specifically B-cell genes like PAX5 and block were mistakenly turned up, and dendritic cell genes like XOR1 and NAO were also turned up.
The cells physically existed, but internally they were confused. Hybrid monsters that didn't know what their job was.
Exactly. The researchers even looked at the wave 2 cells from the fetal liver around day 13.5 and found they completely lacked Zeb2 expression and utterly failed to mature into functional macrophages. The entire standard system was collapsing.
But then, and this is the crazy part, they looked at the microglia inside the brain, and the microglia were completely perfectly fine.
It's wild. The genetic receipts for the microglia showed almost zero changes.
I think there were just tiny blips, like a gene called STMM2 going up slightly and H1F2 going down.
Right, incredibly minor fluctuations. But their core identity, their functional blueprint, was completely intact and untouched by the loss of the enhancer switch.
It's a stunning finding. It proved definitively that primitive hematopoiesis, that very first wave in the yolk sac, does not rely on the same genetic grid as the rest of the body.
It does not.
If we go back to our analogy, it's exactly like cutting the main power line to a sprawling city. The downtown commercial district goes completely dark, the suburbs descend into chaos. But you look up, and one isolated walled fortress on a hill, the brain stays fully lit. And it stays lit because it clearly has its own secret off-the-grid generator.
That's a great way to put it.
All right, time for our second quick recap. Bullet 1. Researchers deleted the -165 kb wall switch in mice. Bullet 2. This caused a total collapse and identity crisis in the adult immune cells and tissue macrophages in the lungs and liver. Bullet 3. The brain's microglia were perfectly fine, proving they don't need that switch at all.
Exactly.
So question for you listening, knowing that the brain has this secret off-the-grid generator, what specific genes do you think are running the backup power?
Well, to find that out, the researchers had to deploy incredibly sophisticated tools. You're looking for a needle in a molecular haystack. So they used a technique called single nuclei multiomic analysis.
Multiomic, so combining multiple methods.
Right. This is brilliant because it allows scientists to combine two different mapping techniques at the exact same time on the exact same individual cell. The first is RNA-seq, which, as we mentioned, reads the receipts.
Right. It tells you which genes are actively being transcribed into working instructions right now.
But the second tool is what really cracks the case, and that is ATAC-seq.
Let's explain ATAC-seq because this is fascinating. Tell us how that works.
So ATAC-seq looks at the physical, three-dimensional structure of the DNA itself. Inside a cell's nucleus, DNA is usually tightly spooled up, almost like a closed book. But when a specific gene is getting ready to be used, the DNA physically unwinds.
It opens up.
Yes, it opens up to allow the cellular machinery to land on it. ATAC-seq maps those open spaces. It tells you exactly which parts of the DNA are physically accessible.
So if RNA-seq shows you what is currently happening, ATAC-seq shows you the potential. It highlights which doors in the biological library are currently unlocked and ready to be pushed open.
That's spot on. And when they ran these algorithms on early fetal cells, looking specifically at the open, unlocked DNA areas surrounding the Zeb2 engine,
What did they find?
they found a massive clue. The DNA landing pads for a specific family of transcription factors called MAF were sitting right there, wide open, in the macrophage clusters.
Wow. So MAF becomes the prime suspect for the off-the-grid generator. But in science, finding a suspect at the scene of the crime doesn't actually prove they did it.
No, it doesn't. You need a timeline to prove causality. So the researchers went back even earlier in the embryonic development to those day 9.5 yolk sac cells.
Right, way back in wave 1.
Exactly. And they found that the Maf gene is expressed, meaning it becomes active, before the Zeb2 engine turns on. It acts upstream.
So it arrives on the scene first, perfectly positioned to be the one flipping the switch.
Yeah.
But they still had to prove it could actually do the job, right?
They did.
Here's where it gets really interesting for you listening. The researchers set up a rescue mission in the lab. They took the mutant fetal cells, the ones that were completely broken and having an identity crisis, because they lacked the main wall switch. And they used retroviruses to essentially hack the cells and artificially force the Maf gene inside them.
Now, just a quick note on that retroviral technique for clarity. It's a classic laboratory method where scientists use the natural ability of a virus to inject genetic material into a host, but they hollow out the harmful viral parts and replace them with the gene they want to study.
Which in this case is Mahos.
Right.
And it worked like absolute magic.
Oh, yeah.
When they forced Maf into those broken fetal cells, it completely restored the Zeb2 engine. The cells stabilized and developed into normal macrophages. It was a total success.
It was a huge success, but biology loves to throw a curveball.
Oh, doesn't it always?
When they tried that exact same rescue trick on the adult bone marrow cells, The wave 3 cells from the mutant mice moth failed completely. It did absolutely nothing to fix them.
Which raises an incredible question. Why would this secret generator work perfectly to rescue a fetal cell, but fail completely when you drop it into an adult cell?
Well, because it turns out MAF doesn't work alone. Transcription factors are highly social molecules. They often form physical partnerships, locking together to bind to the DNA effectively.
Yeah.
The fetal cells already had MAF's secret partner naturally floating around inside them, but the adult cells did not. The researchers realized they were looking for a two-key system. Like a nuclear launch sequence, one key just wasn't enough. MAF needed an accomplice.
And they found the accomplice. It's a gene called Jun, right? Jun, which is part of a larger family of transcription factors known as AP1.
Yes. Adult bone marrow cells don't naturally express Jun. So the scientists went back to the drawing board, they built a dual retroviral system, and they artificially injected both Maf and Jun into the broken adult bone marrow cells at the exact same time.
And that was the aha moment. When Maf and Jun were introduced together, they completely rescued the Zeb2 engine and restored normal macrophage development in the adult cell.
They did. They completely, totally bypassed the need for the missing -165 kb wall switch.
That is incredible. And I know the researchers tested other potential partners to be thorough, right?
They did. They tried a gene called FUS, but it didn't work at all. They tried JinB, and it only partially worked. The exact specific combination lock required both Maf and Jun.
So we need a memory hook for this. For you listening, just remember, Maf and Jun get the job done.
I like that.
Maf and Jun get the job done. They are the secret off-the-grid generator. Together, they form a unique combination code that powers up the essential Zeb2 engine, allowing the brain's very first immune cells to survive and mature before the adult machinery is even built.
It perfectly explains the paradox we started with. It shows the incredible, layered, evolutionary complexity of how our bodies are built, utilizing entirely different developmental pathways depending on the embryonic timeline.
Okay, time for recap number 3. Bullet 1. Using ATAC-seq, scientists found the DNA doors for the MAF gene were wide open early on. Bullet 2. Forcing MAF into broken fetal cells fixed them, but failed in adult cells. Bullet 3. MAF needed a partner, Jun, to get the job done and bypass the missing switch. So, a quick question for the listener. How incredible is it that the exact same gene, Zeb2, is controlled by entirely different keys, depending on what day of embryonic development the cell is born? Biology is just, it's wild.
It really is. But, the researchers at Washington University who did this study are incredibly rigorous. Finding the generator and proving it works in a petri dish wasn't enough. They had to perform one final, ultimate test to prove this happens in a living mammal.
Right, because if Maf and Jun are truly the exclusive backup power source, keeping the brain's microglia alive during that critical first wave, then removing them completely from the equation should cause a total irreversible blackout in the brain.
Precisely. So they utilized another genetic model, which is a mouse with germline MAF deficiency.
Germline deficiency, meaning the mouse is genetically engineered to completely lack the MAF gene from the exact moment of conception. It never exists.
Correct.
Now, from a research perspective, I imagine this is really difficult because lacking the MAF gene entirely is actually fatal to the embryo later in development, right?
All right.
Around day 15.5.
Yes, it's fatal. So the researchers had to look extremely early at days 11.5 and 12.5 to observe the initial fallout before the embryo failed.
And what did they see?
The fallout was staggering. When they examined the embryos lacking MAF, the entire first wave of primitive yolk sac macrophages was simply gone.
Wow, just gone.
Vanished. And when they looked specifically inside the developing brain vault, the microglia were completely missing. Even the specialized macrophages that live in the protective linings of the brain, known as border-associated macrophages, or BAMs, they were wiped out.
So the brain's primary security force simply did not exist. The vault was empty.
Exactly.
But nature famously hates a vacuum. The brain is sending out distress signals, realizing it has no immune protection, no specialized gardeners to prune its neural networks. So the body panics. And this is where the paper reveals a massive biological plot twist.
What's fascinating here is that because the original custom-built microglia failed to develop, the body forced a backup plan. It sent in the second string.
Wave 2.
Yes. It recruited wave 2, those unspecialized monocytes, from the fetal liver. It sent them invading into the brain to try and fill the empty space and compensate for the missing microglia.
Yeah, and the researchers could track this invasion in real time, right? Because these replacement cells are genetically different, they literally light up differently on laboratory scanners. The scientists watched these uninvited replacement cells invading the functional tissue of the brain. They were expressing basic macrophage markers, showing up as bright CD45 positive cells in the brain parenchyma, proving they were trying to do the job.
They were trying, yes.
But they were undeniably not the original primitive microglia.
They were imposters. Well-meaning imposters, but imposters nonetheless.
So let's really think about the implications of this. I want to use an analogy. Let's say I'm building a highly complex, state-of-the-art smart home. I hire a highly specialized custom electrician, that's our microglia, but he doesn't show up. So in a panic, I hire a heavy-duty commercial industrial electrician as a backup or wave 2 replacements.
Okay, I follow you.
Now, the industrial electrician is going to step in, look at the blueprints, and connect the wires. Power will flow. But the lingering question is it going to work right? Does it really matter if the custom expert is replaced by a generalist? I mean, isn't an immune cell just an immune cell? It eats dead debris and fights infection, right?
That is the million-dollar question for the future of neurology. And based on everything this paper uncovered, the answer is an emphatic yes. It matters immensely.
An immune cell is not just an immune cell.
Absolutely not. This entire study proves that these replacement cells have totally different foundational wiring. Because they don't have that early Maf and Jun magic, they are forced to rely on completely different genetic enhancers, like CEBP factors, to function.
They don't have the original blueprint, so they are essentially improvising inside the most complex organ in the body.
Exactly, and because their genetic architecture is different, they might react entirely differently to metabolic stress. They might be too aggressive or too passive when it comes to pruning delicate neural synapses during childhood development. Having generalized replacements instead of custom-built microglia might fundamentally alter how the brain functions, ages, and protects itself over a lifetime.
Wow. Okay, let's do our final recap for you listening, tying it all together. Bullet 1. Deleting Maf completely wipes out the original microglia, proving Maf is the essential key. Bullet 2, in response, the body panics and sends in replacement monocytes from wave 2. Bullet 3, these replacements are genetically different from true microglia and wire themselves differently.
Perfect summary.
So our final subtopic question to you is, if our brain's original custom-built immune cells are lost, can a generalized replacement ever truly do the exact same job?
It's unlikely.
Which brings us to a massive, real-world implication for everyone listening. When we look at this incredible journey we've taken today, discovering the 3 waves of cellular birth, finding the missing -165 kb wall switch, and uncovering the Maf and Jun get the job done secret generator, it's easy to just view it as a brilliant piece of biological detective work.
It is brilliant, but if we connect this to the bigger picture, it is so much more than academic trivia.
Right.
It is the foundation for the next generation of human medicine. Understanding these unique genetic blueprints is crucial if we ever want to repair the brain when things go wrong.
Let's tie this directly to the real world. Think about devastating neurodegenerative diseases like Alzheimer's or Parkinson's. In these diseases, the original microglia in the brain become exhausted, toxic, or dysfunctional. They stop cleaning up the plaques and tangles, and they actually start damaging the neurons.
Which is devastating.
Right now, a lot of cutting-edge medical research is looking at how to cure these diseases by replacing those failing microglia with adult stem cells, meaning taking fresh cells from that definitive 3rd wave in the patient's own bone marrow and transplanting them into the brain.
Which sounds like a logical solution on the surface.
It does, but based on the deep dive we just took into the immunity paper, think about the massive hurdle we now know exists. If we use adult stem cells to replace brain cells, they are missing the primitive blueprints.
Yes.
They don't have that Maf and Jun off-the-grid generator. They are reliant on the adult wall switch.
Exactly. They are fundamentally different cells.
So the provocative question we want to leave you with today is this. If we pump adult stem cells into an Alzheimer's patient's brain, will those cells actually be able to protect the neural networks the way our original microglia did? Can an industrial electrician safely rewire a delicate smart home? Or, to truly cure these devastating diseases, do we need to figure out a way to artificially recreate that primitive wave 1 magic in a laboratory, custom building the exact cells the brain expects?
It's a profound question.
It really is a profound question about the future of healing the human mind, and it's something for you to explore on your own. Thank you so much for joining us on this deep dive.